on the custom parser doesn't make a lot of sense without understanding the domain that this is targeting. And so thought I'd take just a quick time to give you an overview of the specific domain. Looks like we have a new somebody joining. That's actually Christian. Christian, if you don't mind muting your mic. Getting a little bit of background noise. But I'll give a quick overview of the digital pathology domain, and then we'll go into the code of this particular custom parser. So for those of you that aren't familiar, anatomic pathology is used in cancer diagnostics, and it's kind of the last stage as a cancer is being graded and a therapy is being prescribed for the patient. Anatomic pathology is a critical part of making that diagnostic work. A key part of anatomic pathology, and I'm just going to burn through this. Feel free to interject or raise your hand if you have questions. Key part of anatomic pathology is taking the tissue that's been surgically removed, either through biopsy or cutting of the tumor, taking that tissue and mounting it on a slide. There's a few steps here like this little workflow image to show the steps. I think I'm actually going to get my pointer going. Let's do a laser pointer. So processing, then embedding of that tissue. Once it comes out of the embedder, it's sliced into very thin layers of tissue, 2 to 3 microns in depth, typically. And then it's put onto a slide, and typically some type of stain. H&E is the most common for initial staining, and then there's a lot of immunohistochemical stains that are added to highlight individual biomarkers that present themselves in the tissue. And then finally, in traditional pathology, that slide is mounted on a microscope, and a pathologist will review that. In a given case, there may be 3 to 23 slides, depending on the body part and the type of cancer that's being reviewed. And you get images like those there. Oh, let's see. And that is the output of the histology process, the surgical pathology slide. Where digital pathology comes in then is taking that slide, all of the work that's done in producing that slide, and putting it into a whole slide image scanner. And this is a very high resolution camera that a robotic arm generally moves the lens over the slide, and it sequentially captures sections of the slide. We're showing here a traditional 1x3 standard slide, but there's all different types of dimensions of slides for mounting whole brain resections, prostate resections. So the size of the slide varies, therefore there's a number of scanners as well that can take different size slides. And ultimately, that digital asset that's created from the slide scanner can be presented to the pathologist. One of the challenges in the domain is a wide variety of scanners. Over the last 15 to 20 years, as the technology has progressed and as pathologists have been attempting to digitize their work, a number of vendors have come into the market. And so each one of them typically will have a different format for packaging up their slides. Although at the center of it is a fairly standard structure that's been agreed upon over the last 20 years, 15 to 20 years, that takes the overall base image and then packages that with progressive layers to enable zooming and navigating through what is a very large image. And I've given a few characteristics. So these are just of your standard 1x3 slides. There's lots of variation here, but these images that are captured on the scanner are often compressed. In the clinical space, at least here in North America, the 1x3 slide, if it's captured at a magnification of 20x, can be between 500 megabytes and 2 gigabytes in size, depending on the amount of tissue on the slide, like I said, the magnification. And then if there's additional layers from the scanner added, what's called a Z-stack, that can expand exponentially, because each of those layers would be an additional 500 to 2 gigabytes. So you get very large images, and then we'll go into some of the other challenges, but the typical size of whole slide image, at least again in this standard 1x3 format, can be 80,000 by 60,000 pixels, and so you can see we're dealing with fairly large sized images. But once those images are produced, a ton of research over the last 15 years on how to use that digital asset to improve healthcare outcomes. There's lots of work being done in image segmentation, scoring of individual biomarkers, extracting features from the overall digital tissue display, and then in the last 3-4 years, quite a bit of work on classifying using machine learning. So that was the kind of the overview of pathology and the state of digital pathology at a really high level. But targeting one particular clinical domain is breast cancer. So breast cancer is graded with there's several different grading scales, but one scale that's often used is the Nottingham Histologic score, and that scale has 3 different components that are used to grade the severity of a instance of breast cancer. So tubular differentiation, nuclear pleomorphism, and then mitotic cell count. Those 3 factors are used by the pathologist to grade the breast cancer, and then that grading is frequently used to determine what type of therapy, whether you need more aggressive surgical removal, you can treat with chemotherapy or radiation. All of that comes into play once the grading of the progress of that particular cancer is evaluated. In fact, if you remember your high school biology, I graduated from high school in Alberta, Canada, and I managed to get one biology course as part of my high school. I haven't done biology since then, but you may recall the phases of mitosis, and I love this visualization just to give a quick illustration of what steps the cell goes through as it is undergoing the mitotic change. And I show that because What the pathologist is trying to do is recognize and determine those stages in this digital representation of the tissue sample. So this is some examples of individual patches out of a whole slide image that are representing mitoses. One of the characteristics of the mitotic count is identifying the number of mitoses that occur within a high-powered field of view. That represents the field of view of the microscope, and what the pathologist actually does is counts the instances of mitoses that they see within the field of view, and then transcribes that into the report. So there are a few challenges for the pathologist in doing this manual counting of mitoses on the whole slide image. They need to determine the region of the slide with the highest mitotic activity. Remember, they're looking at very high magnification on this tissue sample, and identifying where on the specimen is the highest area of mitotic activity can be a challenge. And then consistently evaluating an indication of mitosis as genuine or lookalike. I know down here there's frequent disagreement between the pathologists on what qualifies as mitoses, and so that can be a challenge in the grading of the tumor. And then accurately counting instances of mitoses in the region of highest activity. So those are some of the challenges. And don't get me wrong, pathologists have been doing this for years. They're very good at it. And so they continue to do this with a high degree of accuracy, but there can be variability. Just to highlight this visually for you on a specific specimen, this is a complete view of the whole slide image, and the pathologist needs to be able to identify where the region of highest activity is. Those green dots represent where the mitotic cell activity is on this specimen, and so their job is to scan through the entire slide on the microscope and determine where that region is. Now, they're trained and they know that certain areas are more likely to have that, the mitoses occurring, but still, this visualization shows the challenge of identifying where the highest concentration may be within the slide. And then they need to differentiate between mitosis and non-mitosis. The blue dots that you're seeing on the specimen now all represent cellular structures that look similar to mitotic activity, but actually aren't. And so that's one of the challenges for the pathologist. And then they need to accurately count the occurrences of mitoses in the field of view that they use. And this gives an illustration of what a typical high-powered field of view would be capturing. So, can machine learning help in this area? That's what the question is, and there's a ton of work being done in this space, and I'm just highlighting a little bit of it. But the approach that most people are taking, and what resonates with me as well, is how can we assist the pathologist in recommending the region of highest mitotic activity And then can we provide a preliminary count of the actual cell mitoses in a high-powered field of view? So we're not eliminating the pathologist at all from the process, but utilizing computer vision tools to assist the pathologist in making their grading. Before I jump into the code, I just want to give acknowledgement to a group of folks that I've been borrowing from and who have done a ton of work in this space. The folks at Friedrich Alexander University in Erlangen, Germany, have done a ton of research and provided some excellent data sets. And if I'm not mistaken, I think Mr. Marzell is in our session this morning. So I'm been grateful for his work and generosity in sharing that at large. But what Christian and Mark and the rest of the team there have produced is one of the largest publicly available data sets of pathologist annotated cell mitoses in canine breast tissue. And you might say, well, gee, canine breast tissue isn't human breast tissue, but there's general agreement that the mitotic structure and activity is similar between these two types of breast tissue, and so it forms an excellent foundation for generating a data set that can be utilized in deep learning experiments. They've also built a really useful whole slide annotation tool and spent a lot of time in addressing the challenge of agreement or disagreement between pathologists in assessing what is a genuine instance of mitotic activity or any type of feature within the overall specimen. And they've built a nice pipeline on Fast AI and PyTorch that they've published their findings for object detection. So I've been borrowing their work, and in fact have been cutting my teeth on machine learning utilizing their work. I've only, I mentioned I've been doing healthcare informatics and medical imaging, primarily in radiology, for the last 20 years, but have just dove into machine learning in the last 7 months. So grateful for the generosity of those that are sharing what they're doing. So some of the technical challenges for mitotic cell detection. I've highlighted this: large images. Again, in the most common format of whole slide images, 500 megabytes to 2 gigabytes. And then very small objects. That's been a theme, and I think we still have a special project in progress for a small object detection. I think mitotic figures qualify as that with depending on the magnification of the tissue. But in this example, tissue at 20x magnification has a structure that's about 4 to 7 micrometers in size. And that patch you're seeing there is a 512 by 512 pixel patch. Additionally, it's a challenge to differentiate between different types of objects within the patch. So highlighting 3 different cell structures. On the left, mitotic lookalike. This has been pathologist annotated as a cell that appears to look like mitoses, but in fact, by their evaluation, is not. Here at the center is a genuine example of mitosis. And then on the right is an example of a tumor cell within this same piece of tissue. So we're not dealing with fridge data set here as far as differentiation between the objects. And that's one of the challenges of the domain as well. Additionally, there's significant domain adaptation required between the processes at each laboratory. I highlighted briefly at the start what the histopathology process looks like, and each of those steps can introduce variability into how the tissue appears to the pathologist. And even more significantly can be the brand and technique that's used for scanning that slide into a whole slide image scanner. Both of those factors introduce a fair amount of variability in the images, and that's one of the components that we need to account for when trying to build a modestly generalizable model for mitotic cell counting. And then highlighted this before as well. There can be frequent discordance between pathologists over what qualifies as mitotic cell division. All right, that was a quick overview of the domain. Any questions so far? Doesn't. Nope. Okay.
Yeah, well,
actually, I do have a question.
Yeah, go ahead.
About the information of the original size of those cells, because here we have just the excerpts. Yeah, so you had said there are 512 by 512 pixels, but do we know what is their actual size? Because I can imagine that could be a valuable information.
Well, it's you can actually get the measurement of the cell, and that varies by cell and tissue type. So it's a translation from what the actual measurement of the tissue is to how it's represented in the image itself, and that varies based on tissue type.
So is it like another layer of the image that is passed to the neural network?
The actual size depends on the, so if it's the digital representation of that cell, that depends on what the pixel, what the actual resolution of the scanner is. And so the size of the image itself, we have as it's recorded by the scanner. And so we're able to kind of translate the resolution into the individual patches.
Because I was imagining once we do the transformation and cut those 5 by 12 patches from the original image, then that information, I was just imagining, could get lost. But so you do have something that to keep it within the image itself, and that information also is included in, let's say, in the input that goes to the neural network.
Yeah, well, and I'll show you that in this data parser. So if you're referring to the original size of the image, yeah, that's one of the pieces of this custom parser that built into the pipeline.
Okay, thank you.
So this is what I'm going to cover here in the next few minutes, loading annotations from the slide runner SQLite database. I'll actually give you a quick demo of slide runner, helpful to understand the types of annotations that we're dealing with, and why I needed a different data parser to manage that. Then we'll select a specific class of annotations to run through the pipeline. We'll generate a slide object that contains some information for creating those patches, and then we'll load that slide object into the IceVision custom parser. And just as a real quick overview, the slide parser passes its information into what I call the FilePatchtoMemory mixin, which inherits from the IceVision parser mixin class, and then FilePatchToRecord mixin inherits from the record mixin. Many of you have built your own custom parsers. This may look familiar, and I'll actually rely on Lucas and Farid to expand on the details of the architecture. I use their assistance in creating this approach, and then we pass the image coordinates and then retrieve the specific patch from the whole slide image to pass into the pipeline. So let me stop presentation here. Move the PowerPoint aside. Are you still able to see my screen?
Yes, yes. Can you increase the font size a little bit? I think you can try Control+. I think that works as well. Yeah.
Is that better?
It's good for me. It's good for everyone else as well. Yeah, this is good now.
All right. So I'll take you to, so I do most of my development in Jupyter and use VS Code to do any coding on the pipeline. This is running on my personal laptop. I use a couple GPUs for actual model building, but do most of my coding on my laptop. Always interested in how other folks are doing it. I'm sure there's a better way, but this is the approach that I'm using. So I'll take you through just real quickly an overview of the notebook that highlights this pipeline, and then we'll step through the functions in a little bit more detail. So one of the areas that I've built into this pipeline is just all of the configuration parameters. Here you can see the various collections of classes. The data set that I'm working on has a number of types of cell structure annotated, and so you can do experiments with more than one class. Interestingly, and I'm not sure if this has been addressed in the IceVision data parser, the only way I could get my class label to display when I had an ID that was greater than 1 or 2 or 3 was to fill in some placeholder values into the IceVision class map. So you see an example of that there. I'm not sure if I was missing something, but that was the approach I was taking to load some of my annotations. And then we have specific pipeline parameters for this instance. There are a number of slides within the data set, and I've highlighted the nature of a whole slide image. Within each of those slides are thousands of annotations, and so thousands of potential patches to be supplied into into the model. So I filter out number of slides depending on if I'm developing or actually doing a real model run. Here we load the database from Slide Runner, and to highlight that, this is probably going to be too small to see as well. I'm not sure if I can expand that. But this table, it's I don't think dbeaver has a ability.
I think it's okay though. I can see it very well.
Okay. So this is the list of classes within this particular data set. You can see a number of cell structures have been annotated, granulocytes, mitotic figures, tumor cells. The pathologist has indicated other more ambiguous cells, binucleated, multinucleated, and then mitotic figure lookalikes. And then I've added another class that I use for extracting larger regions of interest from the whole slide images. So this is where classes are maintained for the data set, and then I'll bring up a view of the of a particular slide within the data set. This is the slide runner tool that I mentioned earlier. Here's an example of canine breast tissue, and you'll see again all of the, in this case, yellow is mitotic activity, and purple is mitotic lookalikes. And so what we're doing in this pipeline is creating a patch for however many of these instances of mitoses that we want to display. And this particular slide was taken at a magnification of 20x. And so this is the view of the slide at 20x. You can hyper magnify, and so we're actually magnifying the pixels at a higher magnification than was actually captured on the scanner. So you'll begin to see some pixelation in the image at that point. But right here at 20x is the actual fidelity of the images that was captured on the scanner. And if you remember that slide I gave you about the structure of a whole slide image, that base layer of the pyramid is what we'll extract the tiles from in our pipeline. So our goal is to create patches of a set dimension. In our case, we're going to do it by 512 by 512 and create a batch of patches to feed into our model. Let me just make sure I've got my size set correctly. Okay. So we load those annotations from the SQLite database, and then once those are retrieved out of the database, we filter those to the selected class that we're creating a data set for, do some randomization of the patches. One of the challenges, like I mentioned, is a need for domain adaptation between types of slides that are colored slightly differently. So can accomplish that by mixing up patches from multiple slides, which do in that cell. And then we load the classes and then load those individual files into those individual patches into the IceVision data parser. So to illustrate this a little bit better, I thought we'd step through the code and let me make sure I have a couple of breakpoints in here. So this is the slide parser routine, and then the record mixin, and then where we actually extract the patch out of the whole slide image. Parser mixin and then the record mixin. And of course, I'm coming up against a memory error, which occurs not too infrequently on this particular laptop. I may have to just step through the code. Manually, which...
Just another question. Is this the file you want to run, or is the other debug one that you have there?
Oh, yeah, actually, thanks, Lucas. You are correct. I'm not even running the right file. There we go. I got my breakpoint on the parser. Thank you. Let's make sure I got a few other breakpoints. Yeah, here we'll collect the annotations. Great. Let's try this one more time. Okay. So right now we are loading the slide annotations from the slide into memory, and so I'm going to just take you through what this looks like and highlights the routine to extract annotations from the slide runner database. And I've mostly borrowed code from Christian's object detection pipeline for pulling those annotations out of slide runner. So we load the slide into memory. Here we determine that we're getting the base level of that image pyramid. Here we are seeing the actual dimensions of that of this particular slide, which is 97,000 pixels by 87,000 pixels. And then if we want to do any down sampling of the image, we can adjust for that as well. And then we go through and actually extract all of the annotations for the entire slide of a specific class. In this case, we're just extracting a single class, which is the mitotic cell class. And then we create the bounding boxes, attach the label for it, and then the slide runner project. Well, this is actually part of the FastAI v1 pipeline. Has a construct of a slide container that embeds the slide path as well as all of the bounding boxes and labels, level, and then our anticipated patch size and width and height. So all that gets created into its own object. So we have the instructions for extracting individual patches now out of that large whole slide image. So I'll go ahead and continue through our routine. Here we are now loading those individual patches into the training and validation record set. So within the slide parser, we've passed those slide container objects into the slide parser. And that's occurring here in the initialization. And then we extract out the level, the width, and the height of each patch as we've set up in our configuration. And then for the bulk of our patches, we actually take a random set of coordinates from within that data set, an X and Y, and search for that particular annotation and get its bounding boxes and labels associated with that particular X and Y position on the whole slide image. So if I describe it another way, we've picked a single X and Y position in that whole slide image, and we know we want, in this case, a 512 by 512 patch, and then we select all of the other annotations that are within that 512 by 512 patch size, and we extract those bounding boxes and labels now into a single patch within the IceVision data parser. Next step is to pass that into the parser mixin. I'm sure there's other ways of implementing this. I'd love to get Christian's take on it as he dives more into IceVision. But the approach that I came up with Lucas's assistance back in December was to pass the slide container object into the record mixin, as well as those the X and Y coordinates. And I will remove the breakpoint from the slide parser so we don't have to look at that throughout the rest of it. Make sure I want to put a breakpoint here now in the record mixin, which we'll get when we visualize this. So the loader is just creating a stub of each of these patches. We'll know the file. We'll know the X and Y position. We'll know the patch size when we try to load that into memory. So to illustrate it, just running the show records method from IceVision, and we'll display a couple of those patches now through the routine. And so the final the method that we utilize is the FilePatchtoMemory record mixin, and we got the coordinates from our original slide parser routine, and in this line, we're actually passing those coordinates now into a method that I have within this helper library that actually goes to OpenSlide, which I didn't talk about and I should have, but OpenSlide is an open source library for manipulating whole slide images, and I'm just scrolling up to that method, get patch. So we invoke OpenSlide to navigate through the whole slide image. OpenSlide can handle the various scanner formats, as well as the metadata that may be associated with that particular whole slide image. And so OpenSlide extracts a patch out of the whole slide image, and then we pass that as an array into IceVision. So that's where the bulk of the work is done in taking the annotations that we had on the whole slide image. We extract a patch out of the image using the OpenSlide library, and now that patch is available for our modeling. The beauty of this approach is you don't have to create the data set at a bunch of different resolutions. You can pick the size and width of your patches for each run and modify that. Initially, when I was using IceVision, I created a routine to extract and then convert to Coco style, and was importing those. But as I was experimenting with different patch sizes, obviously that becomes onerous. And we're dealing with really large data sets as well. I mentioned there are hundreds of thousands of annotations in this particular data set from the folks at FAU. And so we have the luxury of having really large numbers of patches and creating those and writing them to file for each iteration is certainly cumbersome. So this approach enables us to pull those out dynamically from the whole slide image based on how we're configuring a particular run. And I'll stop there and jump back to our notebook. And that's what the output looks like in show record. So here is a sample of 6 patches, 512. Well, actually, I think these are 256 by 256 that I'm showing here. But that's what the patches that we'll feed now into the model. Using just the standard transforms, I haven't, at least in the IceVision pipeline, done a ton with the augmentations library yet, but doing a little bit of resizing and shifting of the images to get some variation, and you can see the impact of those transforms on the individual patches. Implemented WANDB into the pipeline, which is a fantastic tool for keeping track of each run. And I like looking at that to see where my highest map scores are for each run. And the rest of this, you guys have probably forgotten more than I know about the art of tuning a model. I'm still a relative neophyte in tuning, but getting better in this particular pipeline. I've had map scores on a data set of 5,000 patches for training and 5,000 patches for validation of about 86% on the validation set. This particular example is just a 50 patch run, so I could show you the highlight of the pipeline. And then generally load some test patches that haven't been part of the training or validation to take a look at the model as well. And let me close this. Finally, just doing some inference work on it. Thought it would be interesting for you to see. Let's see if I can. This is one of the regions of interest that I created on the slide. These green squares represent a region of interest that I've extracted out of the slide and taken the ground truth annotations and rendered those as opposed to the predicted mitotic activity. The things that I'm working on is how we visualize this within the digital pathology platform that our customers utilize. And so just imposed a grid that highlights the actual patch size that we're doing inference on for this region of interest. And we are at the top of the hour. Hopefully that was interesting. I'm not sure many of you have done your own work in creating a custom data parser, but that's the work that I've done in taking the pipeline that the folks at Friedrich Alexander have created and transforming that into an IceVision pipeline. I've done a number of experiments running different backbones as a result, and one of the fantastic aspects of IceVision is this flexibility. Currently getting best results with efficient det, but also run faster-rcnn and then a RetNet pipeline as well. Looking forward to experimenting as we add additional backbones into the framework. Any questions? And Fareed and Lucas, if you guys can, any questions about the details of the architecture, hopefully you guys can contribute as well.
Yeah, just at the very end there on the inference image, you have some percentage showing on each class. Is that the confidence level or something else?
Yeah, just the confidence level.
All right, yeah.
Where is my, oh, here we go. Yeah, just that's the confidence level coming out of the inference.
And this is for the model that you just used 50, as you're saying?
Oh no, this isn't for that. This is that model that I got about 86% map score on.
I see. Very nice.
That model with 50, it did not do very well at all. I can show you. Here's ground truth. Here's prediction.
It's not finding much stuff.
Yeah, yeah.
And is this like, that is doing 86? Is it doing like a good job there on the image? And of course, I don't know how good the results are there. Does it make sense on the 86%?
Well, it's there's a fair number of false positives when I do it on the broader region of interest. So haven't solved. I need to do some padding of patches. We're missing them on the margins of the patch occasionally, and identifying some that aren't genuine. So it's transposing it into the actual whole slide image. Need to do some work in that area, but on the validation set itself, it's done pretty well.
All right, thank you. Any other questions, anyone? I don't think so. Farid?
No, it's not me.
All right. So I think this is it. Yeah.
Go ahead, Bronzy. What was your question?
Oh, Bronzy has a question. Oh, sorry. I think I muted him in the beginning. Let me unmute. Yeah, now you're muted. You can speak. Sorry.
And second of all, if you are doing your training, do you select patches where normally?
Go ahead, Bronzy. I think we've sorted out the technical.
Can you hear me?
I can hear you now. Yes, I was not able to hear.
Yeah. Okay. First of all, again, thank you for this very nice presentation. And second, if you're training your network, do you just sample from region with mitotic figures, or did you sample from the whole slide image?
Bronzy, you broke up a fair bit there. Didn't get the start of what you were saying.
The question is, are you sampling just from mitotic figures or from the complete whole slide image? Because that's quite important for training for mitotic figures.
For this illustration, just the mitotic figures.
Okay, but in general, you would definitely, you would sample from the complete image, because then you would get better results. Yeah, on your validation set, I would say. I would think so.
Okay, that's great. Bronzy is one of the contributors to the data set, so that's invaluable insight. Thank you.
You're welcome. Thank you again for this really nice presentation.
So I had a couple of questions. Am I hearable?
Yes.
Yeah. Hey, so yeah, really fun, great stuff, and really nice to see. So I just a couple of things. One was you mentioned efficient det. Did you see a really noticeable difference between efficient det and some of the other mechanisms? And that might be an interesting thing to, for folks to think about, especially some of the newer, the recent inclusion of the MMDET models. There might be one or two in there that would be even better suited for lots of small objects. Be interested if anyone has a thought about that.
Yeah, so in my approach, I was getting much better results with fairly minimal additional tuning using efficient det with the map metric as my primary metric.
So was that 80 versus 86 or 82 versus 86? What sort of differences were you seeing there?
One more time, Adam.
What were the mean average precision numbers that you were seeing? What kinds of differences?
So at 2,500 patches with, well, actually, I could bring up the 1 dB illustration. That might take me a second. I was getting about 40.40 with faster-rcnn, and the best score I got with FDET was 0.86.
Cool.
So it's a huge
And-
difference.
Yeah, so that's
Yeah.
a pretty big difference. Yeah, and interesting to think through that. I don't know the efficient det behavior as well as some of the others. And I had just a little question about your picking up of the annotations associated with your tiles.
Okay.
So, does your database have a implementation of that, or did you just scan through them all, or were you using a quad tree or something to efficiently pick up the annotations that associate with the tile?
Yeah, we scanned through all of the annotations on the whole slide.
And other thing about training, so I didn't quite hear whether you were picking random patches from an image, or whether you were effectively tiling it and then picking random tiles that didn't overlap.
Random patches from the image is the approach I've been using. So
that sounds good.
yeah. So,
Yeah, so that
depending
gives you
on how many slides loading, if it's all 32 from this particular data set, I then all of those get loaded, and then we select the number of patches that we want to feed into the model off of that.
Cool. So you basically have effectively an infinite training set there.
Yeah, that's one of the great aspects of this approach.
May I have a question as well, Adam? Oh, okay. I wanted to ask you which MAP was that value for, because there's a number of those. Is it like the IOU 0.50 or the averaged IOU?
Just the default MAP that we use in the coco-metric in IceVision. I believe that's that is at 0.50.
Yeah, it's the average one.
Yeah.
Mm-hmm.
Actually, it's the average of the 10 thresholds from 0.5 to 0.95. So we average on the 10th.
I think so. Yeah, that's the first record that comes from the coco-metric. It's the average.
Yeah.
Correct.
Usually Usually 0.5 give you a higher scores, but if you want to compare, then you should use the mean.
Because 0.86 for the averaged map, sometimes it's called the coco map. It's like basically solved, I'd say. 0.86, that's
it's quite high.
It's
quite good.
Yeah, it's like you could consider the task solved. But yeah, apart from for the false positives, but that's the problem with the metric itself. It's biased in this sense.
But one thing that you have to take into account with the average precision is the fact that you have high scores for the large objects, and then it drops very dramatically with the small boxes. So if you have boxes that are less than 32 by 32, they're considered as small, then your average will be a lot lower. And this is maybe what explains that in some cases you are not going to see some boxes will be not detected, you see.
Yeah, I'm in the process now of creating a routine to generate an F1 score for this data set in the IceVision pipeline, because I've been having the challenge of understanding why I'm getting such a variation between the map score and real world inference. So working on an F1 scoring routine for this.
Do you have an idea on the size of the boxes? What size would be?
The boxes I've done a little experimentation. They're all 50 pixels in size.
Okay. So that might explain why you don't see them, because those sizes, when you have boxes that are between 32 and 96 pixels, they are considered as a medium size, and the average precision for the medium size boxes usually it's lower than the big ones.
Oh, let me rephrase. Or you're referring to the bounding box size. I'm just using the default bounding box size in the framework.
Yes, that's the anchor boxes. The anchor boxes are also the fixed. But what I was referring to is the real size of the mitotic cells. So those boxes, what size are they?
50.
Or 50. So it's considered as a medium size boxes. That might explain why you don't see them. That even if you have like a high score, as the map is 86, it might be that the precision of the medium size boxes is lower than that. I don't know if it's more clear, my explanation, or more confusion.
Well, I need to. That's a great call. I need to run a couple experiments with different sized boxes. I haven't done that in a few weeks,
but maybe you're talking about the shape of the image that you transform those patches to, like the 512. Because the boxes, they're fixed. I mean, the anchor boxes, because that's the configuration of the model. But the bounding boxes or your targets, that's the size of the mitotic cells. Yeah, if I'm calling it correctly. But I just wanted to say one thing. Actually, you have all those metrics monitored. If you use the coco metric, you can. I also saw that you're using WAND, WANDB. So you can, if you're logging your metrics to the WANDB, they should all be there. So maybe you could just switch to monitoring like the medium size MAP or even the small size. I think that's all already being calculated. That's like a default in coco metric. So just maybe check it. You can simply tell the coco metric when you define it to summary equals true, and I think then you will print out the entire summary of all the metrics that it calculates. And one other thing, it's for the efficient det. If you look on the ROSys implementation, there's the configuration files for all different type of backbones that they have, and you could maybe try with like a larger size of your images, because for the most efficient of the efficient dets, the one that has the highest MAPs on Coco, they actually use a pretty large pre-shaping for the images, which is 1280 per 1280. So maybe, I know that's like computationally more challenging, but possibly can give you better results. But you're using FastAI. Okay, I know. You're using FastAI, that's why.
So like what Potipot said, that you're going to have all metrics, we only have that for Lightning. We still have to implement on FastAI to show all the metrics as well. But what you can do, as he's saying, when you define the metric, you say print summary equals true, and then a table is printed. But the logging for FastAI on 1DB, with without the metrics is like still implementation we have to do.
But you can try doing that manually maybe, and also put a PR, because that's be useful for everyone, I guess.
Just to clarify what I was saying, is I'm sharing the screen just from one of the paper, and it gives an idea very quickly about the different precision for different sizes of boxes. So usually what we see is the AP. The AP is the average of all the precisions from 0.5, so 50%, to 0.95, so 95%. So even if you see the mean average at 36, for example, here for faster-rcnn, if you check the same precision score, you will see that it drops dramatically for small boxes and for medium size boxes. So it might explain what you're experiencing there.
Okay, yeah, and you've linked that in the forum already,
Yeah.
right?
Mm-hmm. Yes, yes. And the other thing, what Paul was explaining, is that in efficient det, so depending on the size of your image, you can change the backbone. This is like the ideal backbones. So for if you have a like for example a size of 512, then you can use D0. If you have 896, you can use D3, and so on and so on. So these are like a recommended backbone and the corresponding input sizes.
Yeah, my recollection is we were getting the best results with the D3 backbone. I didn't realize there was so the connection to the input size. That's because I'm actually doing, I think, 768 when getting that that particular score.
Yeah, so I guess you can get good results, and this means that just it's like the recommended one. It doesn't mean that if I use D3 with 68, I'm not going to have a good good result. But these are like based on their experiments that they recommend these sizes. So
cool, that's helpful.
I was thinking about the augmentation, that it might be helpful to use augmentation to help with the domain adaptation from different scanners.
Yes.
Would you think it can help to experiment with like different data augmentation there?
Yep, and Christian's pipeline, the RetNet pipeline, they do a bit more augmentation. And there are other papers where color augmentation is added to address the domain changes between scanners and histopathology labs. I haven't seen anything that's eliminated the need to at least retrain on the particular scanner and the particular slides coming out of a lab, but we haven't gotten to that stage yet.
Okay.
Any other questions or thoughts?
I was wondering about the color. The color of the images here is predominantly red. Is it like the original color, or has it been just colored by some color map?
So yeah, this is the original color of the slide. This is H&E stain, which is the most common stain that's applied to tissue in the histopathology process.
And so images coming from different devices should have the same color?
Well, they will not. There'll be variation in that. And in fact, I should bring up, if I can, I can't find the paper. Yeah, here. Let me bring up this paper here.
But generally, if the color is not so important, one could experiment with grayscale or maybe like contrast and lightning augmentations.
That's not the one I was looking for. So color it generally impacts it significantly, and in fact, what others have seen, even within the same data set, if they were scanned on different scanners, had significant variation in accuracy. So one of the examples for Christian's data set, the canine mammary cancer samples were scanned on a similar scanner to one of the human breast tissue samples, and got much closer accuracy. But any of the human breast tissue samples that were scanned on a different type of scanner had far lower accuracy. So yeah, even the scanner type impacts the data set, and there's folks that are doing work in that right now on the best approach to work with that, but that's still kind of an open domain.
I have a little question. If I understand well, so basically the radiologists, what they do is to identify the region of interest that have like a mitotic how do you say presence, and then they go and kind of label them and then count them. Is it accurate to
yeah,
to
so
say
so one of the tasks of the pathologist, I worked in radiology for several years, and so pathologists would not like to be confused with the radiologist.
Oh my God! Yes, this is what I did now.
Both do diagnostic work. So the pathologist, he is scanning over this entire slide, and they've spent years practicing. They do it very rapidly, and he knows that certain types of tissue within this specimen are likely to have mitotic activity. Like he knows these white sections here at the top aren't likely to have that. So he already knows where to start looking, and then he is scanning typically at full magnification, then across that image. And if he sees an area that he thinks has more activity, he'll focus on counting it there. Honestly, I don't know how much time a pathologist in a clinical setting that's trying to read 70 cases in a day takes in making sure they found the highest concentration. A lot of times, in my experience anyway, they have an instinct about the case, and they've already looked at other biomarkers or seen other tests, and so they're now confirming that with their mitotic count. Now, I'm not saying that that's good clinical practice, but that frequently happens. I'll give you what, if you're still interested, Fareed. Here's an illustration of a typical breast case.
Mm-hmm.
And I'm sure this is the GESTALT image viewer now. So this is a commercial clinical browser-based image viewer. You can see
your product.
Correct. Yeah.
Okay.
So this is the H&E. This is a breast needle core biopsy, which is often used in diagnosing human breast cancer as well. And we have the H&E stain here, and then this is a PR, which is a progesterone receptor staining, and then there's an estrogen receptor staining. And so the pathologist has already looked at the coloration of these other biomarkers, the ER and PR, and is able to identify the region of highest activity, or at least be guided towards it.
So I think that probably at a certain point, like a multitask pipeline would be very helpful in this area, in your domain, in the sense that you can train, for example, a classification and an object detection at the same time, because you can, for example, classify the regions of interest and then train at the same time those same regions with the object detections.
Yep, and that pipeline is something that the folks at FAU are doing and have published on. This is the chart from one of their most recent papers, where they were doing an initial object detection pipeline and then tuning that model with a classification.
Okay.
With some classification training, and on a single, so the AMIDA test here represents a data set that was consistently was used on the same scanner. So, and I don't know if Christian's still on. Looks like he's dropped off, but they created the model on the canine mammary cancer data set, and then they tuned it using a second stage classification, and then they ran inference on this human breast data set, which happened to be scanned on the same type of scanner that they scanned their training set on, and got this improvement off of their dual stage. So went from 0.72 to 0.73.
Cool. So I don't know if you're aware, but we have a project where we are trying to figure out how we can have a multitask pipeline. So Rahul is experimenting with the classification and object detection, and we are trying to figure out how we can merge these kinds of tasks and be able to train at the same time the two tasks, because it's reported that it's very beneficial to take advantage of the training both tasks at the same time. So it might be of interest for you once we figure out all the mechanics to be able to have that pipeline in work.
Yeah, that's great. That's why I'm here. All the development and all the excitement and innovation is why I've been trying to do work with this framework. So I'm you guys have forgotten more than I know about machine learning and been learning a lot by trying out your code.
Thanks a lot,
Well,
Brian. Really kind words.
Cool. Well,
Alright, yeah.
again, thanks, Fareed and Lucas, for the opportunity to share a little bit of what I've been working on, and appreciate all your help over the last several weeks as I've been experimenting with IceVision. And you know, maybe when I get an F1 score set up for this pipeline, we can consider doing another follow-up presentation or something
that
could
would be
deal
great.
more on the metrics. I haven't been doing much experimentation with other backbones just because I'm not confident in my metric yet for comparing. So that's what I've been working on.
Yeah, thank you, Brian, actually, for trying IceVision out, having this presentation, and also contributing. So thanks a lot. Very, very nice.